Our next lymphatic organ is the spleen. The spleen is said to be about the size of a fist, and it fits up against the back wall of your abdominal cavity in the upper left side, up against the diaphragm a bit, and the lower portion of it is kind of up in front of the kidney, so the kidney would be tucked behind that. This portion up here would lie in contact with the stomach on its front face, and down here a loop of the intestine, the large intestine, would be up against this portion of the spleen. So that's the location of the spleen in the body cavity. It is covered by a mesothelial membrane. It is attached to mesenteries in the body cavity, and it has an outer covering here, a capsule that is a dense connective tissue with fibroblasts, but it also contains myofibroblasts, a special kind of fibroblast that has a contractile quality like a smooth muscle cell. So it's a little bit different than some other cell types. And those myofibroblasts can make collagen and do contribute to the collagen that makes the capsule surrounding the spleen. There's one point on the spleen where vessels come and go. So you've got arteries coming in here. So there's a splenic artery. Splenic artery comes in and branches out through the tissue. I'll just draw some of the branches in here to give a little bit of the flavor of what's going on inside the spleen. So you have branches of the major artery. They follow the extensions of connective tissue from the hilum out through the material of the spleen. So there are what we call trabeculae again. We've seen that term before. Connective tissue plates or processes that extend out into the body of the spleen from the capsule. So they kind of follow inward rather than coming much in from the outside. They follow these vessels through here, these trabeculae in the spleen. So these would be sometimes referred to as trabecular arteries. More often we hear about the trabecular veins that head back the other way. So we have this connection point called a hilum or hilus, just this general region here. So the artery and vein are attached here. There's at least one lymph vessel or some lymph vessels that come out from the spleen here, and a nerve supply to the spleen as well. So nerves and vessels attach here at the hilum, which is kind of in the middle of it along this side, along the medial side of the spleen. The rest of the spleen inside here is made up of two different kinds of what we call pulp. There's a red pulp and a white pulp. The white pulp is called white pulp because in a fresh cut spleen, there's less blood in those areas. It's made mostly of lymphocytes, densely packed, without many blood cells in there. The kind of just a lighter color, not as red. The blood and the rest of the spleen makes up what we call the red pulp, or contributes to the color of red pulp. So the white pulp and red pulp make up this here, and it looks kind of marbled or mottled in throughout that if you look at a fresh cut piece of spleen. In our microscope slides, the white pulp, with all of its lymphocytes, will stay in a dark blue purple color, and then the red pulp will still have that more red blood cells in it, so it'll stay in the pink color of the red blood cells a little bit more than what you see in the white pulp. So our white pulp is going to be blue and our red pulp is still going to be red in our microscope slides. Now these vessels, the arteries as they come in here, let me try to make a little drawing down here. The arteries that come in are going to eventually branch into what we call central arteries. So these central arteries are going to be surrounded by lymphocytes making up that white pulp of material. So there'll be a lot of lymphocytes around here. And also in this region of white pulp we're going to see some lymphatic nodules. So you'll see a lymphatic nodule and its germinal center. Some of those scattered around in this region of white pulp surrounding the arteries, the central arteries. So we have a couple of central arteries surrounded by white pulp here. As they travel on and go beyond the white pulp, they form what are called penicillar arterioles, and those penicillar arterioles show it branching a little bit. Eventually form capillaries that just dump the blood into areas of the red pulp we call the splenic cords. Splenic cords. Splenic cords are surrounded by reticular cells and reticular fibers, and they form a meshwork of reticular fibers out here that will contain not only the reticular cells that make the fibers, but also a number of different macrophage cell types, dendritic cells, various white blood cells, lots of lymphocytes still in that. But anyway, the red blood cells will be dumped into here with all the other blood, and they kind of filter their way through to a location that, let's see here, spaces in here. I'll draw it in blue. It's not very good to draw a blood vessel here in blue. It's not really needing to be in blue. But anyway, these larger spaces that eventually converge to form splenic veins. So we have a number of these large spaces out here gathering up the blood that's been dumped into the splenic cords. And these are called splenic sinuses. These vessels here. They're going to be retrieving the red blood cells going through there. So anyway, we have blood moving through what we call central arteries. They move into smaller vessels we call penicillar arteries. Or arterioles. And they eventually feed into small capillaries that dump their blood into this region we call the splenic cord. The blood then traveling through the splenic cord is going to be able to enter very freely into these spaces we call splenic sinuses. And that blood eventually makes its way back out of the spleen along splenic veins. Many of these splenic veins, again, follow trabeculae, so they're known as trabecular veins, but eventually they gather up into a vein that leaves the spleen. There's your splenic vein coming out. So you have this network of the arteries do not parallel the veins exactly in the spleen. They have different routes, so you have the arteries going in and feeding the white pulp, passing through the white pulp, dumping their blood into the red pulp, and then that blood that's been dumped into the red pulp gathers into the splenic sinuses and eventually into these trabecular veins that lead back into the splenic vein, leaving the spleen here. So we need to talk a little more in detail about what's going on here in the white pulp and in the red pulp of the spleen. We'll do that next. Here we have a sample of cross-section of a region of white pulp within the spleen. So there's a central artery, and I've drawn it probably a little too big in here. Probably should be a little smaller than what I have here. But anyway, there's a central artery, and it's not exactly central in the white pulp region. It's usually a little bit off-center. In here we also, let me label that, central artery. In that white pulp, we'll also sometimes see a lymphatic nodule. But most of this is just an accumulation of lymphocytes. Likely there are some reticular cells with reticular fibers kind of forming some bit of a framework within this tissue, but they're not really very visible because of the abundance of lymphocytes. So they're packed tight with lymphocytes. The region, in addition to being called white pulp, is sometimes referred to as a periarterial lymphatic sheath. Or PLS, periarterial lymphatic sheath. That's a region of white pulp, and within it we have the lymphocytes, lymph node, all that going on around that artery. Some of the artery branches that come from this will move through to the region at the edges between the white pulp and the red pulp, and out here on the margin we call this the marginal zone. Marginal zone at the perimeter of the white pulp where you're getting into the red pulp out there at the edge. Sometimes vessels will extend from the central artery on through here and dump their blood into vessels at the marginal zone, vessels of the red pulp out here, some of those splenic sinus vessels. But the spleen is better known for having the blood pass through the white pulp and then enter into those penicillar arterioles, then into capillaries. These capillaries are going to be sheathed with macrophage cells typically, and they call them sheathed capillaries, that terminate out there in the splenic cords beyond the white pulp into the red pulp. This is what we'll look for under the microscope in lab-defined white pulp. We'll be looking for central arteries surrounded by masses of white blood cells, these lymphocytes and dense aggregations, and we'll see those lymphatic nodules within that. So those few things are what we'll look for there. We'll know it as both white pulp, and we'll describe it as the periarterial lymphatic sheath. I'm beginning a drawing of the red pulp of the spleen, and I put these two images, this kind of circle of nuclei and this longitudinal view of nuclei, around these spaces, these splenic sinus spaces, to kind of get us started to know what we're talking about. The cells that make up the wall of the splenic sinuses are endothelial cells, but they're not ordinary endothelial cells. If you were to see the shape of the endothelial cell, you'd see it's a very elongated kind of cell, and there's gaps between the cells all around the margin of the splenic sinus area. So we'd have, like, that would be surrounded by a cell, that would be surrounded by a cell. In cross-section, there's not much beyond the nucleus in a cross-section of these endothelial cells that line the sinuses. And there are significant gaps between the cells, not like the continuous vessels, continuous walls we see on most blood vessels here. So a lot of gaps, a lot of spaces between these elongated, narrow cells. So the back wall of that would consist, you'd have a lot of these long, narrow cells pointed at the ends of those endothelial cells, something like this. And they're all kind of loosely joined together. There's a, instead of having a normal basement membrane like you would find under the endothelium of most blood vessels, there's a very loose network of endothelial material that wraps around here. And let me put that in orange. That's a good color to use it in. I'll use it here. So anyway, there would be a bit of basement membrane out here, but it's discontinuous in the cross section and in a longitudinal section. It would make bands, very loose bands of basement membrane material that kind of surround and loop around the splenic sinuses. So you would find red blood cells and white blood cells, lymphocytes and things passing through these splenic sinuses, mostly red blood cells. So if you get a specimen under the microscope, if it was a specimen where the blood was pooled up in that specimen, you would have that full of red blood cells for the most part. If it had been drained of blood, the organ, before the tissue samples were made, you would have an empty cavity or an empty space inside the splenic sinus. Now, outside of the splenic sinus, we have the splenic cord material. Now, part of the splenic cord material is going to show reticular cells that produce reticular fibers. So you'll have a reticular fiber network out here in the perimeter. So all this reticular fiber region made from reticular cells makes up most of your splenic cord out here. So I'll just put a little bit of splenic cord material out there. And like I said, we're going to have all kinds of cells, and some of the cells are going to be reticular cells. So these, I guess I'll use this pale red marker that I've got here to represent some reticular cells. Those will be reticular cells that produce reticular fiber. Beyond that, there are going to be a lot of red blood cells. There are going to be different kinds of macrophage cells, neutrophils, and lots of lymphocytes out there. So we're going to find our lymphocytes in fair numbers out here. And I can't draw it all in, but you kind of get the picture. Splenic cords consist of reticular fibers and cells along with a number of red blood cell types, white blood cell types and red blood cells scattered through it. Let's make a bunch of little red circles to represent red blood cells out here. Remember, this blood that's coming into the splenic cord was dumped there by the small capillaries that were at the end of our penicillar arterioles after passing through the white pulp. So red blood cells, lymphocytes, macrophage cells, reticular cells, neutrophils, different kinds of white blood cell derived tissues out there making up that pulp out here. So those are the things you'll look for in the slide for red pulp. You'll be looking for these special endothelial cells. Go ahead and label this, huh? They have gaps between them. They're not tight together, as we saw here. They'll be at the perimeter of your splenic sinus, this splenic sinus. This part here would be our splenic cord material. This was basement membrane that we had looped around it. I should say basal lamina to be more correct. Basal lamina. It's produced by the endothelial cell, that epithelial cell. So there's a basal lamina material looped around the splenic sinuses. So next we'll talk about function. When we think about the function of the spleen, we kind of divide the functions into those that have to do with the immune response and those that have to do with blood, different blood functions, I guess we would say. So first of all, the immune functions. As blood passes through the spleen, very much like what we saw with a lymph node, where lymph passes through a lymph node, and we see this kind of filtration action there. We have the same thing going on with the blood here in the spleen. So it's as the blood moves through the spleen, we have the ability to detect any foreign antigens, foreign materials, and as those antigens come into contact with certain cell types that can recognize foreign material, those cells start a process of antigen presentation to the white blood cells, the lymphocytes in there, that will initiate an immune response. So in addition to the recognition process, antigen presentation and recognition, we also see proliferation of B lymphocytes Now, the B lymphocytes produce the antibodies, and the antibodies, well, the B lymphocytes are found primarily in the lymphatic nodules within the spleen. T lymphocytes make up most of the remainder of that white pulp, that aggregation of lymphocytes around the periarterial lymphatic sheath. So we have the presentation, we have the proliferation of those things. Of course, since you're producing the lymphocytes, you have antibody production. And some of these active things actually remove antigens from the blood, digest and dispose of removal/disposal of foreign material. Whether it be a bacterium or some other kind of foreign material that comes in contact with the tissues there. So those are the immune type functions of the spleen. Now, in addition to that, one of the primary things is removal of old blood cells, red blood cells, and platelets. We also see the recycling of hemoglobin. The constituent parts of the red blood cell are broken down by macrophages, and then some of that is going to be processed by macrophages as heme components that are either retained or released in bile, or globular components that are going to be recycled and put into more hemoglobin later. So anyway, we have recycling of those components that are derived from the red blood cells in the spleen. I'll add in here fetal blood formation before our bones have the ability to produce our blood cells, before hematopoiesis begins in our bone marrow. some of our other tissues of the body are actively involved, liver and spleen specifically, in the formation of blood. So fetal blood formation is an important part. In some animals, the spleen is known to have a blood storage advantage where they can accumulate a fair amount of blood within the spleen, and if they have a blood loss, they can see a contraction of the spleen to push that blood out to replace. It's like their own transfusion kind of effect in the body, but that's not been demonstrated in humans. So the blood reservoir factor is not really that important to human physiology. Now, a word about circulation within the spleen. We've already talked about most of this, so just to recap what's going on here. We have one splenic artery that comes into our spleen, supplying the blood, branches out, forming our larger artery branches within the spleen. These feed into those central arteries that pass through the white pulp. So we have our central arteries that go through. Some of the smaller vessels, arterioles that branch off of that, go directly toward the perimeter, the marginal zone, but we're mostly concerned with what goes on as far as going straight through the penicillar arteries or arterioles that go into our sheathed capillaries at the end. And this is what dumps into the splenic cords. Splenic cord really isn't a vessel, is it? So all these up here, these are arterial. I'll put a little red bracket around these up here. So these are arterial branches going in. And then the splenic cord is that reticular fiber and reticular mesh of tissue that the blood cells have to kind of filter through, and then beyond that, those blood cells move into those splenic sinuses. The splenic sinuses then move into trabecular veins and eventually into the splenic vein and out of the spleen. So that's your blood pathway, the path of blood within the spleen. Now, we mentioned that there are some lymphatic vessels in there. They come generally out of that white pulp region and go directly opposite the direction of these arteries. So they would be going from the central artery, penicillar artery, these areas right here. They'd be working their way the reverse direction of these arteries and accumulating lymph. But it doesn't produce a large amount of lymph. It's a rather minor amount, but it does move some of those proliferating lymphocytes from the white pulp back into this general circulation.